Hi, everyone, and welcome to day 2, Biochemistry and the Cell lecture. At this point, you've made it through the first day of the class, and if it felt like kind of a lot, that's because when you do a dissection along with a lecture and then that drag and drop activity and our meeting, we had a pretty full day yesterday, but I saw some really great work done, some really beautiful dissections done. Those mice actually turned out to be, I think, pretty good. In your pictures, I could definitely visualize the organs. So hopefully you got something out of that. You do not have any labs to do today, so you most likely will get the work done a lot quicker than you did yesterday. If you want to take your time, obviously you are more than welcome to do that because they're not due until 8 p.m. So if anyone wants to nap or take long breaks or something like that, but just wanted you to know that it'll be a little different from yesterday because it is all computer work. Today, I want to talk to you about biochemistry and the parts of the cell. So biochemistry is going to involve organic compounds, and we have 4 organic compounds that we're going to discuss: carbohydrates, proteins, lipids, and nucleic acids. And what makes something organic is that it contains carbon, hydrogen, and oxygen, mostly carbon, hydrogen, and oxygen. So when you go to the grocery store and you get organic food, I mean, obviously that organic like vegetables are going to contain carbon, hydrogen, and oxygen, but it's kind of a misleading word. What they mean is that there's not pesticides used. What we mean in this class is it is a compound that's made of mostly carbon, hydrogen, and oxygen, and that is going to include carbohydrates, lipids, proteins, and nucleic acids. So I'm just going to run through each one of those. And then we have ATP. I am not going to talk about ATP in this class. Sorry, I keep going crazy with my screen. I'm not going to talk about ATP in this class. That's more of a cell biology type thing. But just so you know, some people do lump ATP in with organic compounds. Don't worry about that for this class, though. So I am going to run through each one of these macromolecules that we listed above. Please know that if you feel overwhelmed, it's probably because you have not taken cell biology yet. In cell biology, we spend a good week, sometimes 2 weeks, on biochemistry and the parts of the cell. So if it feels like it's a lot, just take your notes, absorb what you can, and just know this will be revisited when you do take cell biology. All right, so carbohydrates, otherwise known as sugars, are going to contain carbon, hydrogen, and oxygen only, and it's going to be in a 1 to 2 to 1 ratio. And so an example of a 1 to 2 to 1 ratio of CHO would be C6H12O6, also known as glucose. So a 1 to 2 to 1 ratio. The function of carbohydrates is fuel for the cell, and it's going to aid in some structure for the cell. And I'll go over that in just a minute. So we know that's fuel for the cell because when we get really, really hungry, that's usually the first thing that we crave is carbohydrates or sugars. Different types of carbohydrates, just some words to describe what carbohydrates are made up of. They could be made up of just a monosaccharide, which is a monomer. And I have some pictures that I'm going to show you in just a second. So let's just go through these words and then I'll show you the pictures. So a mono meaning one, saccharide meaning sugar. It means it's a carbohydrate and it's one ring. And when we say ring in biochemistry, we mean either like a hexagon or a pentagon. And I'm going to show you a picture of that in just a second. A disaccharide, di meaning two, saccharide meaning sugar, means double sugar. So it would be 2 rings bonded together. And those are some examples. So you might recognize glucose bonded to fructose. So glucose is a monosaccharide, fructose is a monosaccharide. Bonded together, they make sucrose. Sucrose is actually table sugar. Lactose and maltose are other examples. I hope that you are kind of seeing the trend that if it's a carbohydrate, it's going to end in OSE. And then a polysaccharide is going to be 3 or more of those rings linked together. And our example for polysaccharides are going to be starch. So starch, when you think of that, think of like a potato. That is actually how energy gets stored for that plant. The potato is actually the energy storage for that plant so that the plant part can actually grow. Glycogen is the energy storage for animals. Now we eat starch and we break down starch for our own energy, but we don't store energy in this form of starch. We store it in the form of glycogen in our liver and in our muscles. All right, so here's some pictures. Hopefully I didn't go too fast through that. So for those that haven't taken cell bio, it might feel kind of fast. For those of you that have taken it, you're like, oh yeah, we did this. So you will do it. I couldn't talk about the cell without talking about the macromolecules. So just take the notes, and then when you do your concept check, it does come straight from the notes. So you'll be fine. All right. Monosaccharides, remember mono meaning one, saccharide meaning sugar. So here are all our example of monosaccharides. We're getting a lot into chemistry right here, but I hope that what you can see is, okay, they're one ring, either a hexagon or a pentagon. Okay, cool, that's a monosaccharide. A disaccharide is when you take two of those rings and you link them together, and this is the one glucose bonded to fructose makes table sugar. You don't really see maltose anywhere, but you do see galactose plus glucose bonded together makes lactose. And all of these are very similar in structure. There's just some differences on how the OHs hang. And that's where we're getting into a lot of chemistry there. But now, so you know, a carbohydrate that has two rings bonded together is a disaccharide. And there's some examples of that. And then finally, our polysaccharides. You can have multiple rings, long chains of these rings bonded together. And your polysaccharides are usually going to be the carbohydrates that are stored for energy. So when you are exercising really hard, let's say you run track, and you're at a track meet and you've been running. I don't know. I was never in track, and I don't think I've ever even gone to a track meet, but I can imagine there's probably running going on. So let's say you are just superstar track star and you're like in all these events, and you do one event, then another event, and you didn't pack your snack and nobody's giving you any food, and you run out of energy in your bloodstream. Your body is then going to access stored sugar, glycogen, that is stored in your liver and in your muscles. I don't know if that was a good example, but I just came up with it off the top of my head. So, yep. So polysaccharides are going to be your glycogen. So for us, I'm only talking about glycogen because this is human anatomy. We store glycogen in our liver. I'm not talking about any other polysaccharides, though. We will save that for biology. Okay, lipids. Lipid is another word for, people usually think of it as fat. It is also made up of carbon, hydrogen, and oxygen. It's also made up of that. And you're like, oh my God, how am I going to tell the difference between a lipid and a carbohydrate? Well, I'm about to show you. All right, so just hang tight. This is another energy storage, but unlike carbohydrates, lipids are going to release energy really slowly. If any of you have ever tried the keto diet, and I like to talk about the keto diet a lot because I attempt to do it often. I'm doing it right now, actually. I've made it 2 weeks. I gained a lot of weight during quarantine. Anyways, you don't need to know about that. But if you ever go on a high fat, low carb diet, you are so tired, at least for the first couple of weeks, because your body does not want to release that stored fat. That is for super, super, super emergency. And so while lipids is energy storage, it is a very slow releasing and kind of a stubborn energy storage, actually. Lipids are also going to be involved in the cell membrane, which I'll talk about in a minute, and there are some hormones that are considered lipids. And so we have different types of lipids here. Triglycerides, that's what you're going to find in food, oils and fats. And you have saturated and unsaturated. So unsaturated is kind of healthy. Saturated fats are going to be solid at room temperature like butter and lard. Unsaturated are going to be liquid at room temperature like olive oil. You have phospholipids, and I'll talk about those in a minute. This is the type of lipid that makes up the cell membrane. You have steroid lipids. Steroids are going to include cholesterol and the sex hormones, testosterone, estrogen, and progesterone. So testosterone, estrogen, and progesterone are actual lipids. But some people might not have known that. And we are not going to worry about this last one. That's lipid involved in blood clotting and stuff like that. Just cross that out. That shouldn't even be on there. Cross that out. We're not even going to look at that. Okay, so how can you tell the difference between a lipid and a carbohydrate? Well, if you look at this picture, I hope you already know they are big in fat, and that's convenient because lipids and triglycerides are fat, and they are like super fat molecules. So you notice there's carbons, hydrogens, and oxygens. It is mostly carbons and hydrogens, less oxygen. So you don't have that 1 to 2 to 1 ratio that you do with carbohydrates. So this is how you would recognize a triglyceride. Super fat. Got these fatty tails right here, long fatty tails. But they are chock full of hydrogen, and when you take biology, you'll find out just how important hydrogen actually is in energy formation, and this is why fat tastes so good. Because we have evolved and adapted to want that hydrogen-rich stuff. All right, this is a phospholipid. Phospholipids are going to have like this phosphate head. And how do we know it's a phosphate? Well, it's the only part that has a P in it, so it must be the phosphate. And it's going to have 2 tails. So 2 tails. So you picture a head that has a phosphate and 2 carbon hydrogen tails. We call those hydrocarbons. Okay, that's your phospholipid, and that's what's going to help make up the cell membrane. And then the third type of lipid is your cholesterol and the sex hormones. These are all, cholesterol and sex hormones are called steroids. So what makes this unique is this, you're like, oh, that's definitely a carbohydrate there. Well, no, because do you see how these 2 hexagons are sharing a side? They're fused, okay? They're fused. And when you look back at the picture of the carbohydrates, those rings are not fused together. They're bonded with an oxygen in the middle. And we'd have to get into a lot of chemistry as to why that makes that a carbohydrate and why that makes this a lipid. Well, I hope you know with lipids are hydrophobic, meaning they don't like water. And so you have lots of carbons and hydrogens here, and they don't like water. So that's what makes this a lipid. And in a biology class, I'd get more into the chemistry of it, but we just don't have the time. I just need you to know that there are different types of macromolecules, and these are the substances or the particular like things like triglycerides, phospholipids, and cholesterol and steroids that fit under each one. I could definitely, if anyone wants me to go further on this, I definitely can. Please let me know, and I will go further on this, but just for the sake of time, I'm not going to, and you don't have to know it. All I would ask you is, hey, where does cholesterol fit under? And you'd pick lipid. So you don't really have to know why, you just need to know it does. All right, nucleic acids. Nucleic acids, now we're going to incorporate some other atoms. It's not just carbon, hydrogen, and oxygen. We now have nitrogen and phosphorus. Nucleic acids are going to be made up of nucleotides. And nucleotides are compounds that have a phosphate, a sugar, and what's called a nitrogenous base. So how can you tell the difference between a nucleic acid and a phospholipid? Well, a phospholipid doesn't have nitrogen. And so that's one way that you can tell. So if you see a molecule that has these atoms, that's going to be a nucleic acid. And nucleic acids are responsible for storing hereditary information. So DNA and RNA are going to fit under nucleic acids. All right, so speaking of DNA and RNA, what are the differences between DNA and RNA? Well, if you just look at the picture, you can tell DNA is double-stranded, RNA is single-stranded. All right. And for those of you that haven't taken cell bio, you're okay. Don't panic right now. I promise we're gonna learn about this. Especially, I think a couple of you are actually taking cell bio with me this summer. Please know that I will keep in mind that some of you haven't taken that class, so you'll be fine. I'm just giving you some information that you can use later in your biology classes. So DNA is double-stranded, RNA is single-stranded. There's also these things called these nitrogenous bases. And so I hope in 7th grade, you guys talked a little bit about DNA, and you talked about adenine pairs with thymine, guanine pairs with cytosine. That's how these 2 strands actually fit together. Every time you see an adenine, it's going to be bonded to a thymine on the other strand. Every time you see a guanine, it's going to be hydrogen bonded to a cytosine on another strand. And that's how DNA gets made. And we would talk all about this structure if it was a biology class, but we're not. We're just kind of looking at it. And so then you notice, okay, cool, here's RNA. Here's my single-stranded RNA. It has adenine, all right? It has guanine. Uracil, what's that? Okay, it also has cytosine. So it looks like the difference is not only is this double-stranded and this one single-stranded, it looks like DNA has thymine and RNA has something called uracil. Awesome. The other differences are DNA is actually going to, for humans, DNA is going to sit in the nucleus and it's going to code for RNA. So that's a good way to remember it. DNA codes for RNA, and then RNA codes for proteins. So DNA codes for RNA, RNA codes for proteins. And they also have different sugars. DNA has a sugar called deoxyribose. RNA has a sugar called ribose. I don't ever really get into that. You know, I never really ask questions on that. I'm just like, yeah, I mean, whatever. They have two different types of sugar. Okay. And then I move on. And then, so if DNA codes to RNA, RNA codes for proteins. We'll talk a little bit about proteins here. I think they are one of the most, I mean, I wouldn't consider them the most important macromolecule. They do the most. I can't say they're the most important because they wouldn't exist if it wasn't for DNA and RNA, but they do the most. So the way you remember if it's a protein is if it has carbon, hydrogen, oxygen, nitrogen, and sulfur. Sulfur should just be included in there because if it has 400 amino acids, it's going to have sulfur. So it's made up of these things called amino acids. And I'm going to show you a picture here in just a minute of what an amino acid would look like. And here are all the functions. Wow, they are so important. They do all these things. They function as enzymes. They can function as hormones. So insulin is an example of a protein, functions as a hormone, meaning it's just a chemical messenger that travels through the blood. Hemoglobin, which is found in red blood cells, transports oxygen. Antibodies. Ooh, we've heard a lot about those lately. Antibodies are proteins. Antibodies are these little Y-shaped proteins that surround pathogens, render them inactive, or tag them for destruction. And they have to fit. So if you've been sick with something, later on you will test positive for the antibodies because your body made these proteins that attack the pathogen. So same thing with coronavirus. They're testing people for antibodies to see if they've had it. Muscles are made out of protein. I think that's the one that we all know because, you know, you got to drink your protein shake before you go to the gym. Muscles are made out of proteins, and collagen and hair. So keratin is support for hair. Collagen is support for skin. Collagen is the protein that breaks down as you age and makes people look old. And that's what people get injected into their face. It's very interesting. All right, structure levels of proteins. So proteins are made up of amino acids linked together with peptide bonds. So what you have here, and I'm not going to ask you guys chemistry questions. I just want to expose you to this if you've never seen it. So amino acids, here's an amino acid, here's an amino acid, here's an amino acid. And remember, amino acids are made up of carbon, hydrogen, oxygen, nitrogen, and some sulfur. So what you're seeing is this structure right here: carbon, hydrogen, nitrogen, oxygen. And then I'm not going to get into the details of what the R group is. Each amino acid has its own little side chain molecule. And I don't want to get into the details of that, but that's what makes each amino acid unique, is a little side chain. If you want extra information on this, I'm happy to send it to you, but I just, we really don't have time to get into it right now. All right, so that's an amino acid, and they're linked together with peptide bonds. All right, and depending on what these R groups are, actually I am going to talk about them for a minute. Depending on what these R groups are, this little chain right here is going to fold in a particular way that gives the protein its structure. So it's going to fold up. So all those amino acids are going to bond together, and the protein's going to fold up into this perfect little ball. All right, so anything that disrupts the folding of the protein is going to mess up the function of that protein. And the things that could do that are like temperature and pH. and stuff like that. So we don't want to misfold the proteins. So proteins are really important. I would just focus on what's the list of functions that a protein does and don't get so caught up in this folding. Just know that it's amino acids that are chained together. Because of the way those amino acids interact with each other, the whole protein is going to fold in a certain way, and that's what gives it its function. So we are going to move on for that. Okay, let's talk about cells. Let's talk about the diversity of cells. Cells are just so cool because look at how different they can look from each other. When you have red blood cells, epithelial, which are like the lining, we'll talk all about those on Friday. Macrophages, which are white blood cells, and nerve cells. Nerve cells are the best. I love them. And then you have your little sperm cells. How cute. So they're very interesting, very different from each other, but they do have some things in common. And here are the things that all animal cells have in common. And so when you look at cell structure, all animal cells are going to have these structures right here. And I am not going to go through all of the functions of each one of these structures. I'm just not. This isn't cell biology, this is anatomy. But just so you know, each one of these structures has a really important function, like the mitochondria is where ATP gets made. The nucleus is where DNA is stored. The ribosomes is where the protein gets made. So DNA is in here, it codes for RNA. RNA leaves the nucleus, goes to the ribosomes. That's where those proteins get made and get folded up and stuff like that. So there's just a lot that happens inside the cell. So all I want you to acknowledge here is even though they look different, even though the epithelial or skin cell looks different from a neuron, they're all going to have these basic features on the inside. Some may have more of one thing than another, like the ER, the endoplasmic reticulum. The endoplasmic reticulum is responsible for the detox of drugs. So where would you find a lot of ER, endoplasmic reticulum? Well, you'd probably would find it in the liver because that is where the detox of drugs and alcohol occurs. So just kind of things like that, but we aren't going to get too much into those. I want to talk a little bit about the plasma membrane though, because the plasma membrane is very relevant to this class because I want to talk about the kidneys tomorrow. Tomorrow. Yes, we're going to talk about the kidneys tomorrow. So let's just kind of do an intro to this plasma membrane. And then tomorrow we're going to talk about transport across the plasma membrane. And that's where kidneys are going to fit in. So I thought it was a very beautiful spot for them to fit in. So the function of the plasma membrane is to enclose the cell contents, to control the exchange of substances in the environment, and it's also responsible for cell communication. It is made up of a lipid bilayer. So remember when I talked about that phospholipid, that thing with the phosphate head and those two fatty acid tails? That's what the lipid bilayer is made up of. The lipid bilayer has cholesterol embedded in it. So can you visualize what cholesterol looks like? If you can't, then can you pause and look back at your notes? What did cholesterol look like? Well, it was a lipid, and we know it's a lipid because we hear about how cholesterol is in eggs, and if you have high cholesterol, don't eat too many eggs, which actually is true to a certain extent, but they can eat eggs, but just not too many. So cholesterol obviously isn't a lipid, it's a lipid. And remember, it has those 4 fused rings. Okay, so now you can kind of visualize it. Glycolipids, you're like, what? So glyco means sugar, lipid is lipid. That's just sugar hanging off of lipids. And then you have proteins embedded in the plasma membrane. And so you have this fluid mosaic model. So here's the phospholipid. So if you can see my cursor, this is the phosphate head. Here's the 2 fatty acid tails hanging off of it. All right. And that makes up this lipid bilayer. The purpose of the lipid bilayer is to make a lipid barrier between 2 aqueous environments. The outside of the cell is aqueous, meaning it's watery, and the inside of the cell is aqueous, meaning it's watery. So if our lipid bilayer wasn't lipid, we would be just a melted puddle on the floor. Anytime we took a shower or bath, we would just dissolve, which would be weird. And then you can see, all right, so that's the lipid bilayer, and then you have proteins embedded. Proteins are embedded. And then right here, you have cholesterol embedded in the lipid bilayer. all right? And then you have all these other crazy things hanging off. I just want to talk a little bit about the phospholipid. So here's the phospholipid again, phosphate head, fatty acid tails. The phosphate head is hydrophilic, meaning that it likes water. It likes water. Why does it like water? Why would a phosphate head like water? Well, water has partial positive and negative charges on it, and what likes negative charges is positive charges. And what likes positive charges is negative charges. And so this negative charge on the phosphate loves the partial positive charge on the water, on the hydrogens of water. And so that's what makes this phosphate head love water, right? The hydrophobic, which is the water-fearing tail. So if you can think of when you have a glass of water and you pour vegetable oil in it, what does that oil do? It separates from the water and it rises to the top. That's what these fatty acid tails are going to do. And the reason is because they're made up of carbons and hydrogens. And carbons and hydrogens, hydrocarbons, they don't have any charges. They don't have partial charges or full charges. They don't want to have anything to do with water because they have nothing in common and there's no positives and negatives that are going to be attracted to each other. That's the most simplistic way that I can explain it. Obviously, with more time, I would go into we would draw pictures of water and the partial positive and partial negative charges. But just know, if it has a charge, it loves water. If it's a hydrocarbon, something that's made out of carbons with hydrogens, it has no charge, it does not like water. So these fatty acid tails are going to turn away towards water, and the phosphate heads are going to be towards water. They're going to be exposed to the water. Hopefully that was a good enough explanation. Like I said, please let me know if you want me to talk about any more of these. I am happy to do that for you individually or for any little groups of you or stuff like that. But in order for us to really get into the bulk or the meat of anatomy and talk about body systems, I do kind of have to go through these kind of quickly. And just so you know, once again, they will be covered in Cell Bio. They will be covered again in AP Bio. And I am not going to ask any difficult questions of you over this stuff. We're just going to move on and start talking about body systems and diseases after we get finished with the cell and the cell membrane. All right, here's another picture of the lipid bilayer. Like I said, phosphate head has a charge, loves water. They love to hang out. Phosphate head down here loves water. These tails are like, get me away from that. And then they kind of crowd in the middle. And so this is what makes the phospholipid bilayer. Water cannot easily pass through here. Okay, it can a little bit, but it cannot easily pass through there. Right, and then you have cholesterol embedded between some of these phospholipids. And what they do is they stabilize the membrane, they maintain fluidity. They just stabilize it. Keep it from getting too packed together, keep it from getting too melty when it gets hot. So just stabilization. All right, and so the last thing I want to talk about are just the proteins that are embedded within the membrane and what they do. And so just so you know, this first one right here, there's proteins embedded within the membrane. They're going to transport things. They can transport ions, transport sugars, transport water, like large amounts of water, okay? Proteins embedded in the membrane can receive chemical messages from other cells. So receive chemical messages. All right? We're going to see this when we look at neurons. All right? There's some that just maintain the cell shape and keep the cell structure. Just maintain the shape, keep it structured. All right? Then there's some that have enzyme activity, and enzymes are responsible for chemical reactions. So there's chemical reactions that occur that involve the enzymes that are embedded in the cell membrane. All right, you have intercellular joining. So the cells of your skin need to have a watertight seal, like a real watertight seal. And so you're going to definitely want those cells to be pressed up against each other with intercellular joining. So proteins help two cells join together. And then lastly, you have cell-to-cell recognition, like ID tags. And so the one thing I want you to know here, because this is going to be important when we talk about blood type, is this right here is a carbohydrate chain hanging off of this protein, a carbohydrate chain. That is the ID tag, this carbohydrate chain that fits into a protein of another cell. So these are two cells joining together, recognizing each other. All right. And when this becomes important is when a person gets a blood transfusion or a transplant of an organ, and you have probably heard compatibility, like the organ wasn't compatible, so they rejected it. Well, here's why. Or the blood transfusion, the blood wasn't compatible, they rejected it. And a lot of times that ends in death, so we got to be real careful with that kind of thing. There's testing that can be done to find out. And then we're going to talk a lot about that when we do blood typing. But this right here, cell-to-cell recognition, is what makes organ transplants so difficult and what makes blood transfusions kind of difficult. You can't just get any type of blood because it is recognized as foreign and the body will reject it. And it rejects it in kind of a violent, crazy way. The immune system will just attack it. And that can lead to lots of problems. All right, that is all I have for you today on biochemistry and parts of the cell. You have a macromolecule food activity. Obviously, macromolecules come from food. So designing food plans and eating plans and menus are actually really important, especially when treating patients who have diseases that are exacerbated by poor diet, like a person with diabetes. And I gave you two choices, a Michael Phelps and a diabetic patient. And I think it's just interesting. So what you're doing is you're helping plan a meal plan for an athlete, and you're also planning a meal plan for or doing a meal plan for someone who has diabetes. So choose one or the other. That's all that's required. If you want to do both, that does get you bonus points. Let me know if you need any help, and I will talk to you guys later.